In today's lecture, we will discuss another phase of cell division in eukaryotes, which is M phase. In previous lecture, we have discussed interphase, and in today's lecture, we will discuss about the M phase. M phase is basically the part of the cell cycle in which the copies of cells chromosome, which is known as the sister chromatids, separates and the cell undergoes division in the process of division. The separation of sister chromatids in the M phase is the critical process that results in a complete set of genetic information for each of the resulting chromosome. Biologists usually divide M phase into 6 stages: the 5 stages of mitosis, which is known as the prophase, prometaphase, metaphase, anaphase, and telophase. And 6th stage is the cytokinesis. It is very important to keep in mind that M phase is a continuous process and its separation into 6 stages is somewhat arbitrary. Now, first phase, which is known as the prophase. That prophase, as the cell enters the prophase, the chromosome becomes visible. As you can see here in the picture, that the chromosomes are visible under light microscope because chromosome was duplicated in the preceding S phase. S phase I have discussed during the process, during the lecture of interphase, that the chromosome will duplicate during the S phase. And chromosome possess 2 chromatids which are attached to the centromere. Here you can see that there are 2 chromatids which are attached at the centromere. The mitotic spindle, which is basically the organized array of microtubules that moves the chromosomes in mitosis, that is formed. Here you can see that the microtubule or mitotic spindles which are formed during the process of during the phase of prophase. In animal cells, the spindle grows out from a pair of centrosomes that migrate to opposite sides of the cells. Within each centrosomes is a special organelle which is known as the centriole, which also is composed of microtubules. Some plant cells do not have centrosomes or centrioles, but they do have the mitotic spindle. So next phase is the prometaphase. Prometaphase is basically the disintegration of nuclear membrane. Here you can see that there is disintegration of nuclear membrane that marks the start of prometaphase. Spindle microtubules, as you can see during the prophase, that they are outside the nuclear membrane. So now as the disintegration of nuclear membrane has taken place, so these centrioles or these microtubules will move towards the nuclear region. The ends of certain microtubules make contact with the chromosome. For each chromosome, a microtubule from one of the centrosomes anchors to the kinetochore of one of the sister chromatids. A microtubule from the opposite centrosomes then attached to the other sister chromatids, and so the chromosomes is anchored to the both of the centrosomes. The microtubules lengthen and shorten. Pushing and pulling occurs during this stage. Some microtubules extend from each centrosomes towards the center of the spindle but do not attach to the chromosome. Next phase is very important phase, that is the metaphase plate. Metaphase. During the metaphase, the chromosomes becomes aligned or arranged in a single plane which is known as the metaphasic plane. As you can see in the figure, this is the metaphasic plane, and between the 2 centrosomes, the centrosomes now as opposite ends of the cells within microtubules radiating towards and meeting outward and meeting in the middle of the cell. Centers at the spindle poles. A spindle assembly checkpoint, which is very important step in this during this phase, that is basically is aligned on the metaphase plane and check that spindle fibers from the opposite poles. The passage of cell through spindle assembly checkpoint depends on tension which is generated at the kinetochore, as the two conjoined chromatids are pulled opposite directions by the spindle fibers. This tension is required for the cells to pass through this spindle assembly checkpoint. If a microtubule attach to one chromatids but not to the other, then there will be no tension which is required for pulling the chromatids, so the cell is unable to progress the next stage of the cell cycle. The spindle assembly checkpoint is basically able to detect a single pair of the chromosome that are not properly attached to the microtubule. The importance of this checkpoint is illustrated by the cell, that if there is any defect, because if there is any abnormality, if there is no separation at this point, then that will lead to the abnormality within the cell.